right, we can start. Welcome, everyone, to this session. My name is Effy Vayena. I'm a professor of bioethics at ETH in Zurich, and I'm very pleased to be here. Thank you for joining this session. I think it's going to be the more exciting session. I know we're in competition with others, but I promise you excitement here. We're here to discuss organoids, and I'm sure by your choice of coming here, you know what these things are. So I'm not going to give you an introduction. We'll have our experts talking more about it. But I think what we would like to do is to ask some questions and debate not only the progress we're making in organoids, but the challenges they pose, not only the technical challenges, the purely scientific challenges, but also the challenges in terms of ethics and society. And of course, beyond that, the potential we have here for governance and regulation. And that is exactly in the mission of GESDA's mandate, which, of course, recognized and identified, rather, not recognized, organoids and the current developments. I don't know if you paid attention in the big room, but we may have also here the teal panel, where organoids, you can see on that circle, they're actually quite close to that center. So we're expecting things to be happening sooner and not in the further out future. So the question we want to discuss is how we're progressing, how we ought to progress, and what can we do in science and diplomacy to enable these technologies to develop while at the same time making sure that we're not violating ethical principles and that you're promoting a version of progress in our society that we agree that is the right version of progress. And for that, very challenging but interesting discussion, we have a fantastic group of experts here with us. I'll start with Alison Motry, who is next to me, who's professor of pediatrics and cellular and molecular medicine at the University of California in San Diego. And our next panelist is Professor Matthias Lutolf. Matthias is Professor of Life Sciences at EPFL here in Lausanne, and Vice President and Scientific Director at the Roche Institute for Translational Bioengineering. And of course, with us, we have Françoise Rivasseau, who is a senior consultant for technology and diplomacy at the World International, at the WIPO. So, what we're going to do before we get into the discussion is to show a little film, a short film that the GESDA team prepared. I think it can get us on a good kind of level for the questions. terrific. And if you can judge from the music, there's a lot of excitement that we're feeling about this whole subject, which is very good, because what you're seeing here, but also I'm sure what you know, is that we do have a lot of hope about how organoids can actually help us have a better understanding about human organs and about disease, but also applications, the ways we can use them in precision medicine, in personalized medicine, and beyond. So let's start first with a question to our experts about whereabout are we? I mean, that's all fascinating, but it was not very specific. Could you maybe, Alison, start us off? Where are we right now in terms of the developments that we're seeing in organoid research first? And then we'll move on to clinical, perhaps, applications.
Yeah, I think on the research side, it really depends on what kind of tissue you're talking about. We, as a scientist, we learn more about certain tissues than others. Of course, there's lots of excitement about the brain, because the brain is one tissue that we don't have immediately access, because it's inside of your skull. So that's what I do. I recreated the brain outside the uterus or outside the skull. There are cancer organoids that you can just take from the cancer and grow them. Heart organoids and lung organoids, for example. Pancreas organoids it's a good one. Until like 2 years ago, we had a beautiful tissue with no insulin. 2 years ago, they produced insulin. For the brain, we also have the cells forming a nice tissue that resemble like a fetal brain. In 2019, we showed that they start connecting and form neural oscillations in the same way that you can record from an EEG in a human brain. So I think on the research side, we are learning how to make those tissues more and more similar to the real thing. It's not yet the real thing. I mean, it's an organoid. It's a miniaturized version. Not all cell types are there. It's not vascularized. There's lots of technical issues that we need to solve. But honestly, I think in the next 5 years, all these technicalities will be solved. Reproducibility, vascularization, size of the tissue, we're going to solve those in the next 5 years. I think that's where we are.
And that's across I mean, you spoke a lot about the brain organoids, but that's across let's say, organs, or wherever we have tried that.
Yeah. I think there are problems that are intrinsic to all organoids. Take vascularization. I think everybody's suffering with this problem now, because we don't know how to stimulate the phase formation inside those tissues yet. And this affects all types of organoids. But apart from that, there are tissues that are easier than others, and I think brain is perhaps one of the most challenges, because there are so many different pieces, and the complexity is it's high compared to heart, for example.
Very good. Let's see, Matthias, that's, let's say, sort of the research. If we try to group them a little bit, because we'll come to the challenges that each group is producing. Where are we in terms of using those more in the translation, clinical translation? Are we anywhere? Like, how are we developing there?
I think we're in an interesting phase now where we see proof of concept for many applications in translation. But it's fair to say that there are major challenges that we need to address by bringing in additional, technologies to solve some of the issues. For me, organoids are already an extremely useful tool for basic biology. I think that's clear. They're really changing the way how biologists work, how life sciences lab operate, and they become almost a commodity. So instead of working with cells in 2D culture or using an animal model, many labs, or most labs around the world, in fact, are using an organoid to address some of their questions. And that's a big impact of this new technology. Now, if we think about, really impacting the life of patients, it's a different story. But I think we're seeing validation that this technology can make a difference in precision medicine, in particular solid cancer, where we can use, patient-derived samples, and we see that they maintain the mutational burden you know, the genetics are really well preserved in cell culture, which is already a big deal if we want to model treatments that are based on chemotherapy, for instance. You know, so we can start to use this tool in very translational areas, which I think is exciting. But we're just at the start of seeing, I think, an explosion of effort into realizing all these exciting applications.
And for example, use in deciding, as you said, the appropriate chemotherapy or appropriate drug treatment, any kind of treatment. Are we still there? We're seeing a lot of potential, but are we're still there also in the beginning, right?
We have, you know, there are, I would say, landmark publications that have made a correlation between clinical responses in patients treated by chemotherapy and ex vivo or in vitro measurements, and there's a very, very good correlation. Now, if we then think about more complex therapeutic modalities like immuno-oncology, where you have antibodies or even using cells, then it becomes more challenging, because in this case, the model needs to replicate not only the tumor that grows, but all the cell types that can affect how the tumor grows, the immune cells, other cell types from the so-called stroma. And if we can't really preserve this communication between these different cell types in the tumor, we can't really model in vitro how these new generations of therapies act in vitro. But this is now work that's ongoing in many cancer centers and academic labs around the world to improve these models to become more realistic.
I'm by training a bioethicist, so I'm listening to you talking about these impressive developments. There's like all kinds of questions in my mind, and also in my field and scholarship about, okay, how do you consent the patients that give you tissue? Where do you stop when you're progressing in the development, or in the case of even clinical translation, right? Okay, who's going to pay for it? Who has access, etc. So I'd like to go into that, but before going into that, I'd like to turn to Françoise a little bit about, we can go do deep dives on these questions, but maybe Françoise could give us a bit of a review, let's say, of where we are at the global level, not in the development of organoids, but rather in in the work, what's happening with those things at the governance level, right? We have them around for a little bit, not too long, but enough to have those questions addressed.
I really believe, thank you very much, and first let me tell you how I'm honored to be with you today. I honestly believe, I'm in charge of artificial intelligence also in WIPO, that IP problems will be the first manifestation of global governance on your discoveries. Because basically you have first to know who knows and is able to do what with the discoveries, and this is precisely what IP tries to solve. And obviously, IP and ethics are very closely linked. It's a two-way street. The ethics and the ethical constraints impact directly the IP solutions, but IP solutions, when they are established, help shaping the ethical concepts also. So the ethical questions that we have, that you know much better than me, but I briefly sum them up because they have a direct impact about the IP solutions we could envisage. First, we don't have a agreed definition of what an organoid is. When I review the literature, I find that almost as many definitions as scientists. Yeah. So we would need a definition. Second, as you mentioned, we need to think what specific ethical protection for cells donors. The consent is very important, but also, and maybe even more for IP, the privacy. What protection for cell receivers? For human, obviously, but maybe we could have some in 25 years other questions, artificial intelligences, animals, chimeras, whatnot. And obviously, for last but not least, our tarte à la crème, would organoids themselves at one point of development require some kind of IP protections? I think particularly to gastruloids and cerebroids. We ignore still what kind of sentience they could possess. I use sentience. I know it's a word that French don't like. But for me, who read science fiction in my youth, I immediately understand what sentience means. So that's the constraint through which the IP solutions have to be envisaged. And then very briefly, what kind of protection? We are here in uncharted territory. So you could think about inventors and patents will be of application. But there are a number of blocks down the road that we would have to remove. A bit the same, by the way, that the blocks we find when dealing with the questions raised by artificial intelligence. And the first one is, would really the classic definition of an inventor applies to what you are doing? First of all, you know, if you want to file a patent, you have to file you have to make available to your IP office the way you have reached to build up your organoids. And maybe the recipes you could try to patent, but the result itself, it's living cell, you know. So it's supposed to develop himself through some natural processes. So here, the patentability of a thing is not obvious, please it or not, to Monsanto. Nature is not so easy to patent. So another problem that I mentioned, which will be very difficult to deal with if you go to patent, is the problem of privacy. If you use the cells, let's say my cells, I could very well claim that my cells, my DNA, cannot be made public as an invention. My DNA is not something you have invented, you know. So how do you solve this issue? It will be a difficulty. And therefore, very likely, we would have different solutions within different jurisdictions, one in China, one in US, one in Europe. Today, for artificial intelligence, they are not unified, and it's in WIPO, we are not yet even authorized to talk about trying to unify the solutions, and should do, but it's divisive to use the word uniformization, so, you know, problem also for that. So if patents are difficult, we could go for trade secrets, which is another way of protecting things. But then we could have a bit the opposite difficulties than those we get with on patents. For example, do you think we can use trade secrets for personal medicine? Well, probably not. You know, you will help your neighbor while repairing, let's say, from your fantastic brain cells, some accidents he had in a car in LA, despite the fact that transport in LA is secure. But anyway, it can happen. And then you leave him going, and then he travels to Japan and needs a doctor to look at it, and then he will say, I'm sorry, I cannot tell you what I have, it's covered by trade secret. No, it's not possible. So trade secret could also pose a serious question, and then you have consequences in terms of ownership, in terms of liabilities, in terms of revenues. And just to conclude, to make you laugh a bit, but if we are going to 25 years in a futurist angle, we could also maybe think, should we not recognize some rights to gastruloids or cerebroids, particularly if they are advanced enough to be phased like that with an artificial intelligence? As you know, just to make you think, there is already one jurisdiction in Australia and one patent law system in South Africa which have accepted artificial intelligence to file patent on their own name. So you see, so basically here you have a lot of difficulties to solve. But it's uncharted territory. It's a great time to start thinking about it. Thank you.
Thank you, Francois. They are not going to solve the problem alone.
No.
I can tell you that. Hopefully not. Because I think we have to solve this problem together, all of us with the different disciplines. But so thank you very much for sort of giving us, through the lens of IP, the many, many layers of challenges we have, that they are not technical. It's not about to vascularize the organoids, which is so important. But I think I may dare say perhaps equally, if more important, if we want this to become something that, again, helps us progress and develop in the right direction. So I'll just try to unpack a few of the issues that you mentioned and maybe discuss with also our practitioners here. So you mentioned the consent as being a problem, not a problem, a challenge, as far as sort of the donors of tissue, the tissue donors are concerned, right? I was going to maybe start with you, Alison. How do you go about that? What do people consent for when you use their tissue?
Yeah, I think we developed probably the first consent form for brain organoids. And well, I mean, we basically explain the research and we go a little bit into the future, what can happen to those cells or those organoids, including becoming conscious. And I had like people decline because of that. But most people would consent on the use of their cells because they see that there's a value for disease modeling, for cure of some neurological conditions that we have no idea how to cure today. So so this is all laid down very specifically in the consent form. And we do our best to kind of predict where this technology will go. But of course, I mean, things might change, and that's where I have like an IRB, that it's a group that reviewed the research, and they might ask from time to tweak that consent form, make it more clear or incorporate new advancements that we have with the technology. So it's a very dynamic process, and sometimes, yeah, we might have to go back and reconsent that family because they use like an old version of a consent form. So that happens as well. But we try to be very specific on what are the cells going to be used. We're going to distribute the cells to the entire world. Some people will become millionaire by finding cures.
You're saying that?
We have to say that.
Okay.
And you're going to have no remuneration to your side. So you agree with that. Your cells will go to space. We're going to treat your cells with psychedelics. And you're going to be OK with that. And most people consent for that.
It sounds very exciting. I mean, you know. So, I'm so, probably the same happens in your research, right? In terms of consent process.
Yes, exactly. So what for us is makes it much easier is if we don't know who the patient is. We're not informed. We cannot trace back to the individual from which we get samples. This makes all the difference for the doctor to share the materials. So
it's tissue that it's taken, but still the patient has to, of course, agree to that.
Agree to that. We are allowed to do sequencing. So we need to understand the mutations, but we do not know who the patient was. But we can work with the material. We can do whatever experiment we want.
Right, so not knowing who the person is protects the patients or the persons who donated the sample. Privacy, of course, but still we have the issue whether, even if my privacy is protected, whether I want anybody to send anything that has something to do with me to space, or to treat anything that has anything to do with me with psychedelic drugs, right? So there's still a sense in which my autonomy cannot be protected just because we are protecting my identity, which is one of our major concerns. So I think what we see is that everywhere we do these kinds of research, in our protocols, there is a consent, obviously. I think there's variation in what kind of consent we have. Some are more specific, some may be a little broader in that sense. But something that you mentioned, I think it was you that mentioned that, Alison, was that we can't always know what's going to happen, is something that limits also a little bit the power of consent, right? It protects us in terms of identity because we'll say, okay, this is not going to be, my DNA is not going to be, or my identity is not going to become public. But in lacking the possibility to predict everything that can happen is, I think, one of the bit of deficits that we have with consent. It can do so much for our ethics, but it cannot do all of our ethics. So that's where we need other layers of looking at, what is exactly that we're doing. So you mentioned IRB or review committees. They don't look just at the consent, so whether I allowed the use of my samples, but also what exactly are you doing? What's the purpose of what we are doing? And I think there we put another layer of protection of the whole outcome of this different kind of research. So with consent, I think it's not perfect, but at least we have some, I'd say, consensus globally that we need to have that process and document. Some places it's just a document, some places it's more of a process where people are more engaged. and that takes care of a certain part. But then, Francois, you mentioned the various models of what we're doing. Essentially, when we're down the line, we want to go on with commercialization, right? Because that's where also partly the IP questions are coming in. Where are we in the global dialogue? I mean, we have identified that we have a problem, or not a problem, we have a challenge, or there will be a challenge. Do we have already a conversation about this, or we're just flying blind?
This is to me to answer?
Yes.
Thank you.
Do we have a global conversation
Well,
here?
indeed, we have a global conversation on AI and other frontiers technologies. So technically speaking, we could inject in this, and this has taken some time to be accepted in WIPO. It has been first an initiative by the former DG, after consulting all the head of agencies in Geneva, you know, because everybody in UN wants to know what will be the rules applying to artificial intelligence. And it took 2 years before member states did accept the fact, because they were very reluctant at the beginning. But now everybody accepts the fact that there are conversations. As I said, we are not yet authorized to push too visibly towards uniformization of the solutions. What we are doing, what WIPO is doing is, because as you know, WIPO is one of the very few UN organizations which has some own money, because it gets some money from the patent, so it finances some solutions for the IP institutions nationally to know what the best practices are, and we hope that through that some kind of common understandings could emerge. But we have not yet begun to discuss particularly organoids in these conversations.
Okay.
Some national intellectual property institutes should take the initiative of asking us to inject that in our IP conversations.
Okay, so
on frontiers technologies.
So we have identified, and that's actually very important for GESDA as well, we have identified an area in a fast-moving field with huge potential. However, a critical piece in the governance of it, which is intellectual property, we're maybe very vaguely...
There is room to receive it, but you have not yet knocked at the door.
We have not even at the door. So then one thing that's going to be happening as well, as you said, since we're not having a global approach to this, it's likely that countries will take that up differently, right? They'll develop their own sort of approach to broader governance of this, including the IP. Yes.
If I may just add another dimension, which is not purely IP, but you know, last year I was still ambassador in Geneva, and I was working with the Human Rights Council. It may well be that there could be some initiative in the Human Rights Council also, given the fundamental human right dimension on some 25 years projections.
Right. Okay.
Maybe another avenue for international governance.
Right, so we have that for sure noted for international governance as well. Let me go back to, maybe back to Matthias. One of the issues that comes up, it's linked to IP, of course, but one of the issues we have with precision medicine is that, although we're very hopeful about it can do for individual patients, we have the big question of access. How can we have these types of particular specific developments that precise and very personalized being accessible by all the people that maybe need them instead of the very few? Are there any thoughts already in that direction that you're aware of in terms of access to this? I know we're a little bit early in the translational pathway, but I'm of the opinion, this anticipatory opinion, that we should be maybe thinking about that a little earlier than at the last minute. So I'm wondering, what's your take on that?
Yeah, I think cancer is a good case to look at. And, you know, patients are now diagnosed by taking biopsies, histological analysis, and genomics is performed. And all it requires is another biopsy on which one can measure how a certain drug or a combination of drug or a certain dose acts on that specific patient's cancer. this is technically a relatively small step. And every patient would immediately agree that, yes, do these experiments, please. The question is, how do you bring this together with creating a business for a company who develops the machine that can actually perform these measurements, because it needs to be very highly standardized. And I think right now what happens is a lot of experimental efforts to do exactly that. So you can, clinicians are collaborating with people who can grow these tissues and do the measurements and now at the stage where most people are is kind of do a retrospective analysis. So you get a piece, the patient is treated, but only after the treatment can you then perform the experiment to see whether it actually whether your model actually works. But I think in the future, we need to really find a model where every patient can benefit from this advance, and it's going to happen. Of course, there are many small companies who want to move into that space because they're developing these machines that can do the measurements. And probably, one technology will win, and then there will be a company that can manufacture and sell these machines, and then they will be used in the clinic, completely independent of the pharma companies. Because the doctor has the whole panel of drugs available, and they want to do combination treatments, because every tumor is different, and every patient's tumor responds in a different way. So to me, it's a game-changing development for this technology, and it's going to happen. I mean, we're setting up exactly these measurements now.
And do you think it will be affordable down the road?
It will be affordable because it's very cheap.
Okay.
It's very cheap. If you look at the costs for society, if you don't treat the patient well, you can neglect it. The technology and what it takes for this product of an in vitro diagnostic tool is nothing compared to the overall treatment. And I think it's a no-brainer, really.
Okay, that sounds. I like the sound of that one. But Francois, you have to say something. You want to say something?
I entirely charmed by what you say, but precisely, I remember what I've learned through IDS crisis. Finally, the drugs are not so expensive, and most recently with COVID-19, there will be very quickly
The drugs are expensive, but not the
diagnostic.
To do the diagnostic test. No, this is a different discussion. The drugs, of course.
Yeah.
The drugs are, of course, very expensive.
So anyway, there will be a huge push to get this
no way around
very quickly.
There is no way around.
The only international institution that I see which could help cannot be Gavi or Global Fund, because their mandate doesn't allow, is Unitaid, which is the institution, the vertical fund which can buy innovative things and which does buy diagnosis system, but it has not many funds. And we shall have also the problem of a patent, to come back to IP, the patent on your machine. And there, we know what had happened during COVID-19. You had a WHO at the center, and then this very complex system of Act A, and then this idea of biobanks and hubs and patent pools. Do you see the problem being much more simple for you than for it was in recent past experiences?
I mean, there will be the question is, should a company do that?
Yeah.
Right?
Yeah,
or should it be another entity that develops this diagnostics platform? Should it be a business? But I would argue, if you have companies who compete to build the best technology, we will be successful. And again, I feel that the diagnostics part of the overall costs for treatment are going to be very small. And that could still be a business for a company. So I'm pretty optimistic there.
Very good. I'll leave the patent question. We'll park it for a little bit. I know it's going to come back. But I wanted to go back to Alison, who is sitting here in the hot seat, because he's working with brain organoids. And everybody gets so excited about brain organoids. Why do we get so excited about those organoids? And you've been quoted in many places talking about them. So tell us a little bit about it.
Yeah.
I mean, as I said, I think from all the tissues in the body, brain is the one that we understand the least. And this is due to inaccessibility very early on in life, so we don't know how the brains form. And also when you're an adult, we don't know how your brain works. So you can do no-invasive methodologies, EEG, MRI, can have like a global view of how the brain works, but you cannot understand its synapses, how each neuron connects to each one, and what goes wrong when you have Parkinson's, Alzheimer's, dementia, all kind of neurological disorders. And that's the reason why we don't have good treatment for those conditions. So the excitement comes from that part. For the first time in the history of science, we have a model, a human model, where you can recapitulate neurodevelopment of a person in the lab in a control situation and do experiments with that tissue. So we never had that before. I mean, it's very different from the cancer. You take the biopsy, you do the test, you know if it's working or not. We never had that opportunity for the brain. Most of the biomedical sciences so far has been relying on animal models, which is, to be honest, I mean, unsatisfactory to understand the human brain. So that's the excitement. That's where the excitement is.
But what are we worried about specifically about those brain organoids?
Yeah, so
that's less worrisome. You know,
are we worried?
Are they mini brains?
Yeah, I think and that's the brain factor, right? I mean, when you talk about other tissue organoids, pancreas, lung, whatever, people don't really care much. I mean, they understand. But when you talk about the brain, the function of the brain is to acquire consciousness about or self-aware. Mm-hmm. And I think there is a yuck factor about, I mean, these organoids reaching that level, which is, my personal opinion, is inevitable. They're going to do that as we develop the technology. And this is the perception of the society. And that perception was not always for the brain. That's why you say, I love you with my heart, not with my brain, because at one point it was the heart, not the brain, the center of emotions. So now we focus too much on the brain. And that's the part that society sees, or lay person will see. If you have now a brain organoid derived from my cells or the cells from someone from my family with a neurological condition, is that brain organoid, when they start to acquire self-aware, is that part of the person? Do they have any rights? How should we protect that kind of science? I mean, how far is too far? What is the limit of this technology? So I think that's the brain factor. So
there, for example, do we have, let's say, if the organoid starts sensing, right, acquiring some sensory type of capacity, do you, in your research or in the research that your colleagues do, have a stop rule or not? Or you keep going and you'll see?
So when I mentioned that at one point we reach an optimization of the protocol, that we noticed that these organoids were not only forming a nice tissue that resembled the human brain, but they were sending neural oscillations that is very similar to an EEG. At that point, I start to worry. And then I connected with the bioethicists from UC San Diego, and we organized a symposium where I invited the philosophers of the mind, the lay people, and many others to debate where we are, what is this technology, and what are the potential consequences for the society. What I had in mind is to prepare the society for what's coming. So I didn't want another Dolly. I mean, if you remember, Dolly the clone just happened and people got by surprise. Nobody knew this was cooking inside the labs until the final notification. So the brain organoid, as I pointed out, is inevitable. It will acquire some self-aware consciousness. And I don't want this to reach the society or to take them by surprise. I want to cook that idea and why we are doing that, explaining all the steps where we are. So as a summary of this meeting, we wrote a paper, a manuscript, proposing to create some specific milestones for the brain organoids where we would eventually pause the research and rediscuss or reassess where we are. And so we develop very specific methods to do with these organoids to see if they are conscious or if they have any level of consciousness where we would increase the discussion. So that's where we are.
And that's, I think, sort of the approach of proceed with caution, right? And collect groups of people, groups of experts that can tell us a little bit, or guide us as to how we're proceeding. Now, that's the approach of your group, and hopefully from others. But perhaps going back to our global thinking here, do you think, Françoise, that we could possibly reach a point where, as a global community of scientists, before we get to IP and all of that, let's say for the specific problems and challenges we have with how far do we go in the development of organoids, right? Do you think that we could get to sort of a global, informal agreement amongst the scientists that will be proceeding in that way? Not regulation, not, you know,
I'm an optimistic, so I would hope that this could happen. But let's say there is also a place where this could be discussed. It has been discussed in the case of AI, where a code of conduct has been elaborated. It's UNESCO in Paris, because it's a place where researchers can meet at global level. But honestly, this code of conduct in terms of AI has little influence on the real development. So I don't know. UNESCO, despite being very impressive in terms, is not very influential on the real life, unfortunately, despite the fact that it is in Paris. When I turn to the real world, I see that the conception between the two sides of the Pacific, between San Diego and Beijing, are quite different. You know, we have seen this precedent of the twins trying to inject to alter the genomes to make somebody more intelligent, and this has been prohibited by China. But I don't know if the US would agree with that. Not necessarily. I think the Chinese position is widely supported outside of the US. But I call that a consequence of a human-centric approach that we have globally. But the reality is that this has not happened, and maybe this could be something that I was discussing about GESDA. I said, you know,
GESDA's homework is the list of things to do for GESDA.
We have today a big risk, geopolitical risk, which is that the science in the world would be cut into segments: the Western segment and the Chinese-Russian segment. Already, CERN is hopefully a counterexample, but we see in many places program of cooperation between China and Russia on one side and the West on the other being slowed down, sometimes interrupted, or sometimes maintained but not renewed.
So that's actually, I mean, again, very much into what GESDA is about, right? Those providing these opportunities for identifying these kinds of issues, that they would be better served if we have global approaches to this, and if we could get, again, multilateralism in action here, because that's, I think, what we're seeing. But as we are, I have one last set of questions for you, and then we can get some questions from people who are here. We are thinking, right, no, we have some more time. We were thinking, you mentioned, for example, the questions about the issues of preparing people to give us, to not freak out, right? If you tell people you're growing organoids, and these organoids can sense pain, and that is what organoids start meaning for the public, maybe this will create a strange response. And often when we have these kinds of things happening in scientific developments, we have either a very reserved public or strict regulation. So, but it's not exactly very productive. So you talked about trying to have a bigger dialogue. So my question to you and Matthias as well is, do we have a public dialogue about those developments, whether they are about how far we can go or what the patients then might be wanting? Are we talking about this with our publics?
Not enough. Not enough. I think before I answer, I want to maybe put things a little bit into perspective in terms of what we can generate with organoids. Because one topic we haven't mentioned is the synthetic embryos.
Yeah. Yeah.
This is a completely different ballgame if we think about societal issues, moral, ethical. This is orders of magnitude of well, we can go far beyond that now, right? I mean, a few weeks ago, 2 groups have generated embryo-like structures that are quite similar to the mouse, using mouse stem cells. And now several groups are working on generating synthetic human embryos in culture. This raises questions we need to discuss here with society. And I think we need to have some rules. I think the scientists they need to have some guidelines, international guidelines in terms of what they can work on and what not. But of course, there is that problem of having, China and Russia. And, it's going to be hard. But I feel the International Society for Stem Cell Biology is doing a really good job in raising all these issues and providing guidelines that can be taken up by the countries to engage in a dialogue with the people and then to come up with rules about this.
And actually, going back to, because you mentioned the gene editing experiment like a couple of years ago, that most of us freaked out because we thought we're not crossing those lines, we're not doing those things. That was the common understanding. And of course, we had this event happening. We had real people being produced out of this. It wasn't just an experiment, even that we are worried about. But what we also saw at the time, I think, is this global community sticking to that. We should condemning that approach. Of course, it happened, and it shouldn't have happened. But we did not see a split. Even China itself cut that and put that person in prison, etc. So we had the global response, and of course, choosing not to be cynical about what people would say goes behind doors, etc. But if we are taking what's happening in the global world, in the global scene at face value, we saw a global coalition, which an agreement amongst researchers. We're stopping at this point. We're not going further. It's wrong to go further. And that's, I think, I would say, of course, now we're debating, we're thinking, we have a number of instruments and groups thinking about gene editing. And I'm thinking whether we need to do the same here. But before we get to an incident like the incident we had in China, we should be proactive here in working with the researchers to get to those codes that you were talking about right before, in the special categories as well. So I'm back to my question about, you talk to the bioethicists, we have this conversation here. With who else? Are we talking with the people, with the publics, and do we understand or communicate with them? What's your take?
I would agree that not enough. I think there is an effort from the International Stem Cell Society, from researchers who are doing that to get the word out. But that also requires help from the media. We have to reach out. We have to do more outreach. And those things are not easily funded. For example, outreach, right? I mean, in US, it's very hard to find funding for outreach. So if I want to produce like a YouTube video explaining my research, I mean, who's going to pay for that? I can't find money. Sometimes I pay myself just to get it done. So this is something that we are paying the price by not communicating with society. And we have the means of doing that. We have all these very nice social media platforms. It's just a matter of creating the language and exposing people to that discussion. So I don't think we're doing enough.
And in your understanding also of engaging with people, is it about telling them what you're doing, or is it also about listening to what they think?
Oh, I think it's a dialogue. I think it has, and that's what we did in this very first meeting. So we had a dialogue, and by the way, this was recorded in YouTube. I highly recommend it because you can see the questions that got from the public. What are the potential side effects of this technology? I mean, can you teach these organoids to control a drone and put bombs everywhere? Was a question from the audience. On the other side, I mean, you have parents with children with severe neurological conditions who are begging, please do not stop, continue. So we have those things that are very emotional in the audience, but people need to understand exactly what's going on, and it needs to be a dialogue, yes.
I was looking at also not on the public engagement activities, because as you say, I think they're not so many to find. So we need to reiterate that and the need for more of that communication. But I was also looking at to see if there are any social science studies trying to study what are people's attitudes about those things. And I have to say, correct me if I'm wrong, that these are scarce as well. Yeah. We don't know, even not from a public engagement perspective, but also from a perspective of studying attitudes and perceptions of people. I came across only one study from Utrecht looking at donors of material which was used for organoid development, and very fascinating things came out of this study, including, for example, the notion of self to something that's of you, even if you have really nothing to do with that or you don't feel anything in a sensory way. But people in that particular group felt that this is something of them. And so how then, when we develop our frameworks, our ethical frameworks, our regulatory frameworks, our IP frameworks, even consider these kinds of sentiment that this particular community had, maybe in other communities is not present, but I think we're seeing also a lack of, I don't want to say interest, but maybe attention in understanding what two people for whom we will be doing the research and who will be the patients and who will be in a democratic society, the people who vote for what kind of society we want, then this is going to happen when we have no idea of what is happening. I don't know if you have any thoughts on that, Françoise, and how do we really understand those things?
I believe you have helped us paving the way for partly my answer when you said, I don't give any money for the time being to those who have cells. The reality is that if I make the parallel with artificial intelligence, you know, who is the one who will benefit from artificial intelligence? Forget about inventors, trade secret, or copyright, without entering into technicalities. Who will get the revenue of that? You could take that it is the one who has designated the algorithm. In that case, the initial algorithm, it's you. It's the donor who has designed his own cells. You know, who owns that. But in artificial intelligence, we all know that the most important thing is how you feel. You feed the artificial intelligence, the one who provides the data. So in that case, it would be probably you who are feeding these cells to make them productive. Or is it finally the one who owns the artificial intelligence, or the one you would sell your organoids to? Or is it the artificial intelligence itself, as I said, in that case, the organoid himself, who will have to get a part of the revenue? Should we go for a shared proportion of revenue to each step of the production, to the cell donor, to the cell receiver, to you, obviously, as the main...
That's quite futuristic, that line of thinking. But I wanted to say that in terms of samples and ownership and how we use biological material from humans, we're not into a vacuum. As we may be, and we don't start from the same point that maybe we start off from for AI, because we have been using human tissue.
Yeah, absolutely.
And we have generated cell lines. We have also scandals and a lot of discussions around how we do this the right way, how we commercialize. So we are not in a vacuum, I think, a complete vacuum. But the question is whether when it comes to
brain,
these, not only to brain, I mean, we have also other challenging categories. When we have this potential that these organoids seem to have, whether we need to refresh those concepts and agreements we have with our patients. I mean, typically we tell the patients, you give your sample, and as you said, we're out, right? We separate the sample from the person as long as we protect their privacy, etc. But in this kind of developments we're seeing here, are these concepts the one that will serve us all the best? And I think that's part of also what we need to be rethinking. Maybe they do, and we don't have to change anything. I'm not saying we do, but I think it's this is where what I find problematic is when we are in this potential, these kinds of developments, that we need to discuss those and listen also and understand the expectations and what people want around those things. And here is where we need to do a little bit more, or quite a lot more, than what I think we're doing. It could be that we can proceed as we are, but from this small study, it seemed that we have to take into account concerns that maybe did not come up when you just give your, a blood sample and people do something with that.
Can I add to that? Because I'm familiar with the work, and when I talked to the authors about that, they said, yeah, I mean, people who donate organs, right? I mean, donate like a heart from your son who passed away. People feel attached to the recipient of the heart, right? They think that it's the heart from their son now. And I think the brain organoid touches this identity part as well. But again, is lack of comprehension of what makes you unique. Because even if I clone you, your entire body right now is not going to be you, because the experience that brain would have is going to be completely different than you. So it's not about the physical matter, and more about the experience. And this is something that we know from neuroscience, but the public is not aware of that.
Exactly, and that's why, and it also has to do with how it's going to be sold to people, right? Because it's very sensational to write somewhere a piece of you is now somewhere else. It's not a piece of you, it's something else. So how we communicate those things, but how prepared we are to listen also, those concerns, because you can address this question better if you know this question exists.
Right.
And I think that's where we tend to have a gap in those more let's say, provocative types of science that we're conducting, which is so important and we should embrace, but with those under certain conditions. So we have another 15 minutes, and I have many more questions, but I think since you're all here, maybe this is an opportunity to ask our esteemed panelists here questions yourself. So if there are any questions in the audience, it's a good time to take them now. I'll start with you. I don't know your name, but please identify yourself. You need a mic for the, yes, thank you. Thank you, Martin.
Thank you very much. My name is Claudia Reinprecht. I'm from the Austrian Ministry for Foreign Affairs. And it's rather a comment on the discussion you started on the global governance discussion and the ethics of it. And Mr. Rivazot has mentioned UNESCO. Indeed, I just wanted to say these kind of ethical discussions on implications of science technology do take place in UNESCO. There's a bioethics committee, and I would recommend you get in touch, because that's exactly what we need. The problem, I think, with the discussions, and I'm very much keen on watching the YouTube video, but it's how do we really get a global discussion in place? And global means not only discussions in US or in Europe, but how do you get the global South involved in these kind of discussions? That's where UNESCO can be an added value. And that could be a question to you. How much have you had opportunity to get in touch with scientists from the global South and other regions? Because these kind of discussions are very much constrained to us. So, well, that's about it, what I wanted to say. Thanks.
Thank you for the question. There are a few questions in that comment, not only one. So, please.
I have friends all over the world, so I travel a lot and I talk to lots of people on the stem cell field. So there are exchanges of communication with several countries about that. And I mean, I think the scientists are more or less on the same page, although the technology is surprising us as well. 5 years ago, I would never say that a brain organoid will have any level of consciousness. Now, I'm more careful. I would say, well, maybe it's going to happen. So we better be prepared for that. So it's changing as we go, and the whole community is learning that as well.
Any other comments in relation to
well, one maybe provocative comment is that the type of science we do here is extremely expensive. I mean, we do experiments like single-cell sequencing where you pay tens of thousands of dollars to make measurements on organoids. And you can wonder whether this type of research is justified given the real problems that we have in this world, right? I mean, that's just one thought here. And the other one, I think there's a lot of diseases that affect the global south, which are rare, that no company wants to work on because it's not a business. But those diseases are extremely interesting for organoids because one can study interactions of the host with parasites, and those really, really interesting science can happen by collaborations with scientists in the global south. It's something that is now starting to happen.
And as some of those diseases do not stay there, as we see,
Yeah.
as we've seen,
Yes,
I think that will motivate us to become a little bit more global than what we have been in that respect. But maybe a quick comment to your point about the role of global governance. At the same time that we saw the UNESCO document coming out on artificial intelligence, and I'm going a little bit off point here, we saw that countries like the EU is trying to regulate this because the code of ethics wasn't sufficient to make the behavior, to enable the behavior we wanted to have. So at the moment, we're moving a little bit from code to regulation, not because one replaces the other, there's a combination, but it seems that at least in some areas of technological development, we go into this natural development from code to something which has somewhat more it's a bit more, has more teeth. Okay, another, Zara, you had a question, yeah.
Thank you. So my question would be to Alison and Matthias. So in 5 or 10 years, all technicalities are solved. You have big, nice, vascularized organoids. They do what they should do. They are sentient if they are brain, or have sensory capacities, let's say, and so forth. So my question is, what would you do with them, and what would you not do with them?
Yeah, so that's a great question. This is my personal opinion. Okay, don't take as a general. Yeah, yeah. I don't distinguish a conscious organoid from a mouse that is also conscious. And I use mouse for my research as well. But I do it following certain rules. For example, I calculate the number of animals that I need for a certain experiment. When I don't need them, I sacrifice them using humanized methods so they don't suffer. There is no pain involved. Most likely, those rules would be applied to the organoids. That's what I would expect the community to embrace. So I would follow the same things. So we just like set a standard procedure on what we can and cannot do it with them. For example, eliciting pain for no purpose, no reason. We don't do that with animals in research. I wouldn't do it with the organoids. But I will use them to answer specific questions regarding the human brain or how to cure neurological conditions. So that's where I am.
So there are many diseases that have no cure. Just one example, inflammatory bowel disease cannot be treated. I would use those models to try to find a way to cure those untreatable diseases, and of course neurodegenerative diseases are falling into that category. So there's a lot of work to be done to develop medicines, therapies, which is very exciting. And you know, kind of coming with my Roche hat on, you know, there are also a lot of tools that are needed to make the process of testing the safety and efficacy of drugs much better. So like having a human liver that's functional would be game-changing, because you could immediately see before you engage in clinical trials that something actually is safe. And you can apply that to all the healthy organs. So there's lots and lots of areas to apply this. What I would not do personally is kind of in this area that I mentioned before of building human embryos. I feel this to me is kind of a red line where I mean, I understand that we do not know much about early human development, and I understand that there's a rationale to use those models to do that. But the question is, how far do you push it? Do you then go to organogenesis? Do you build the whole thing? Because there are questions everywhere you could address with this tool. And just as a personal decision for me, this is not an area that I would move into in terms of the human models, actually.
Can I ask you a question? What would be the decision point for a human blastocyst? How would you define when you're crossing the board?
Yeah, I mean, now we have as a guideline day 14, right? So it's sort of gastrulation when that starts, but that's also somewhat arbitrary, right? You could argue, you push it further until the CNS develops and the entity experiences pain. That would be sort of when you stop. It's a tricky one. I don't have an answer, really. I feel that this question needs to be revisited now with the day 14 rule, because there are these new tools now. You don't need to have a fertilized egg to do such experiments.
And there is conversation about that rule.
Of course,
yes,
of
already.
course. And that's an important discussion that needs to happen right now. The other area which I would not touch is making chimeras with embryos. To me, I mean, I feel there are more interesting questions to address, but maybe you have a different perspective on that one. But that's just...
Okay, Sarah, you have another question, yes. Exactly. And there's another, to have two questions in the back, yeah.
Exactly on chimeras, so I mean human animal.
Yeah.
Organoids or whatever. Sorry. Chimeras, human animal. The question to you, François, is the patent system, WIPO or offices, could they decide that they would never patent chimeras? Just say, I put a red line there. So then you remove commercial incentives or stuff. Could that be one way you make red lines in futuristic science?
I think this would require a decision which, given WIPO ways of working, to be implemented strictly, should be unanimous or quasi-unanimous. I doubt that WIPO would be the first UN body to generate such a decision. But that's a personal view that WIPO maybe doesn't share, and I just talk in my pure personal capacity.
But it's an avenue to explore, I'm guessing. We have two questions in the back. Whoever gets the mic first.
Okay, thank you. Kind of one comment and two quick questions. So I just want to go back to a point that Effy made earlier about there are existing kind of ethical discussions from the long history of biology and genetics and mechanisms that we can look back to. And this reminds me of Dr. Gill's comment this morning about learning from history of the science. There are these existing things. For example, the WHO also convened an expert group on genome editing recently. This was after the He incident, but still, there are international mechanisms for some kind of multilateral conversations about this. One question for Matthias. So you went on the issue of kind of equitable accessibility. You were kind of optimistic that technology will become so cheap that clinics will easily use this, but I guess the counterexample is genome sequencing after 20 plus years is still not largely not used in a lot of clinics. So are you particularly optimistic about
well,
technology?
of course, yeah, it's a really good point. Genomics, I think, as a tool, is still unsuccessful with most patients who have cancer. We just can't predict how a drug whether it will have any effect, right? Because we lack biomarkers. So let's say if only 10% or 20% of a patient cohort benefit, then you wonder, is it really worth? But if you had a tool, and this requires a lot of R&D to really build the tool to be functional, but if you had the tool to grow and maintain the patient's tumor functional in a dish, and you could treat it, it works. It will work. I mean, this this is a 10-year horizon type of effort. But if it works, there will be so much pressure. You're obliged to use it. I think it's going to happen. It's going to happen. It's just that right now the tools we have available are not good enough. Just work for a few, a certain percentage of patients.
Thank you. And on a question of public engagement, Kevin Esfel, who works on a lot of gene drive research, for example, and public engagement on that research, I remember he once said something along the lines of, it's not true public engagement if we don't accept no as an answer. So would you be prepared to accept no as an answer when engaging with the public on going forward with some of the more controversial aspects of this research?
It's me.
Anybody who'd like to take that question?
Yeah, I would. Yeah, I mean, yeah. In Switzerland, I want to stay here because it has a lot of benefits. It's a great place to do research in Switzerland. And if the law would change in ways that would affect the program that I have, I would then adhere to that. Yes, I would not move to other places because of that. There's so many interesting things to do.
Okay, so they can take no for an answer. We have one more question.
Hi, I'm Eloise. I'm part of the youth cohort people. So yeah, but Kelly, in response to your comment on how we can globally educate people on organoids, I would say one of the best ways to do so would be to incorporate this research and to incorporate this concept into school curriculums, into our biology classes. For me, like as soon as I learn something interesting, I want to go straight home and tell my parents about it or tell my friends about it. And in this way, at least the conversation is started. People are talking about it. And furthermore, like by teaching the following generations about what type of society or humanity that they're stepping into once they're done with their schooling, they'll be prepared for this. They know what they're going to be actively confronted by these decisions, by these issues, and if in their schooling in high school and university, if they're able to understand these concepts before they're confronted with the issue, as you guys were saying, then the conversation is just going to grow, and when people are interested in things, they talk about it. So I think that would be the most efficient, but obviously there are many other ways. But then my actual question is, what was the most difficult part in the development and research? What would you guys say is the most difficult part in the development and research of organoids?
From the technical side?
I guess in any way, technical or social or economic, any way.
I think right now probably one of the technical things that we are facing is reproducibility and teaching people how to do it. We have robust protocols, but they are not perfect. So the organoids are not identical to each other. So this is something that we're still trying to figure out why. So the organoid, I mean, as a concept, the cells self-organize into a tissue. And for some reason that we don't fully understand, I mean, some organoids develop slightly different than others. So it might be like the nature of the things, but ideally for the science to advance, we want protocols that work in everybody's hands. So that's a technical issue that we're facing now.
So is the reliability of the production?
Yeah.
Thank you.
All right, thank you very much. I think we are at the end. Do we have one more minute or not? Martin, one more minute. So just so we don't end very abruptly, I just want to give you a few seconds to each of you to answer in a tweet length type of answer. What is your wish from your perspective in terms of frameworks for continuing the work with organoids? If you had one thing, the most urgent thing that you think it needs to be done in terms of frameworks and governance, what would that be? I'm going to start with François because he's working on frameworks and governance.
Some regulation, I have to say, because that's without that we shall not be able to address seriously the issue. It can be a soft regulation, like those coming from societies, or better regulation.
Matthias?
Yeah, it's a tricky one. I think to make sure organoids are openly accessible as a technology for whatever people want to do, that they become like a commodity in research, but also in how companies operate. I feel it would be really terrible if you would need a lot of money to access it. You know, this I think is a very important thing to consider.
Access, right. Alison?
Time.
Time?
Time.
To do more work?
Yeah. The technology takes time. And similar to other, I'll give you like examples of stem cell related technologies. Blood transfusion, right? I mean, people would die from that. It took like several years, and it's still not 100% successful in blood transfusion. So science takes time to refine the technology. And I think we need time for proof of principle. As soon as we start curing people, finding cures, I think then the society would embrace the technology. But the scientists need time to do that.
Okay, so we have to give you some time. Meanwhile, we'll be working on frameworks, and I think the message here is that this is extremely promising. It is not far away in the future, although we need more time to get this to be better, but we should be working on frameworks that will enable this technology to be accessible, to move towards a direction that we can all share the benefit that it can produce. And with that, thank you for being here, and thank you, our panelists, for taking the time to have this conversation with us. So join me to thank our panelists for this discussion.